After we saw the protein correlations, now it's time to go back to RNA and try to plot the UMAP. But before we do that, we need to process the RNA first. A UMAP, or a uniform manifold approximation and projection, is taking highly complex data with thousands of variables. In our case, our RNA matrix is sparse, right? Lots of zeros, and it's large. It's 4,000 by 2,000. And you only want, let's say, the top 50 components. And it takes this complex data, high dimension, and simplifies it into a 2D or 3D visual map. So with visualization, it will group similar data points into clusters and separate the similar ones. So to plot this UMAP, first we need to filter some genes and then take the highly variable ones and do a PCA on it. So we can reduce the dimension and map it. So I'm going to define a function here, preprocess RNA for UMAP. And it's going to take RNA as an argument, which is another annotated data, and output another and data. Now I'm going to make a copy of our input. Next, I'm going to remove genes that are detected in fewer than 3 cells. For example, imagine a gene appears in only 1 cell out of 5,000 cells. That gene is probably too rare to be useful for clustering or visualization. And I would like to normalize so that every cell has the same total RNA count. In this case, 10,000. And we're gonna apply log transformation, as it will compress large values and makes the data easier for PCA. Now we want to find the most variable genes across cells. Let's say the top 2,000. These are called highly variable genes. So the idea is not all genes are useful for distinguishing cell types. To do that, we're going to use an algorithm called SORAT, which does not compare all genes to each other. It compares each gene only to genes with similar expression levels. This will fix the problem that when genes with high counts always win, just because they're big and have big variance, this SORAT will fix that. So let's say if you have gene A with mean of 1,000 and variance of 5,000, this will win. But if you have a gene that has a mean of 5 and variance of 800, this will lose unfairly. And I'm going to make a new AND-data object containing only the highly variable genes. Now we will standardize each gene so it has similar scale. In fact, PCA is sensitive to scale. if one gene has much larger values than others, it can dominate the PCA. Now let's use scanpytl.pca to reduce the RNA data from many genes into fewer summary access called principal components. And we're going to use a solver called ARPack, which is a mathematical library being used under the hood to solve this PCA computation. So PCA needs to find the direction of maximum variance in your data. Mathematically, this means solving an ion wave problem on a huge matrix. And it's best for the sparse data, large matrices, when number of components is way less than number of features. There are other solvers, such as randomized, which has faster approximation, good for very large dataset, and auto and full. So now let's build a nearest neighbor graph between cells. Doing this, we are finding out for each cell which 15 cells are most similar to it. So using the first 30 PCs to find the 15 nearest neighbors for every cell. Now let's run UMAP. After this line, each cell gets two coordinates, UMAP1 and UMAP2. After this, we're going to run the Leiden clustering, which uses the neighbor graph to group similar cells into clusters. It's going to add cluster labels into RNA.obs Leiden, so you can access it from there. Now we're going to return RNA high variable genes. Now that we have the preprocessed RNA, we're going to define this function plot RNA UMAP. It's going to take the RNA high variables as input. And so first we have to call the preprocess function within this, and we will assign it to a variable. Now we plot the UMAP. This shows whether cells naturally cluster based on RNA. Now for the argument color, we're going to pass Leiden to tell it to use Leiden cluster labels to color each cell. So as I said before, Leiden is using neighbor graph to do the clustering versus using raw expression matrix. And for example, if we want to compare it with the k-means, in k-means we say I want exactly k clusters. K-means tries to force all cells into 5 groups. But Leiden is more like, I'm going to see how many I will find. So in single cell data, you usually don't know the true number of cell types in advance. Also, k-means prefers round ball-shaped clusters, but biological cell populations are often irregular, curved, or connected by transition. So k-means are good when clusters are round and you know how many you want. Leiden is better when clusters are graph-shaped and k is unknown. Now let's run and see this plot. Okay, I think I imported plot RNA UMAP from the wrong module, and it was stored in RNA UMAP. That looks good. Okay, so let's look at the plot. First, each dot is one cell. Each color is one Leiden cluster. The algorithms found 9 clusters totally. So we have 3 major separate regions: the left blob, the middle one, and the far right. The fact that these regions are physically separated on the UMAP is important. It means the cells in different regions are very different from each other biologically, not just slightly different. So the actual physical distance shows real biological distance as well. The fact that we have 3 well-separated blobs with visible white space between them shows that we have 3 major cell lineage, to the point where even after compressing 20,000 dimension to 2, they are still far apart. This is a good sign for our model. The RNA space has clear structure and major cell type boundaries are unambiguous. The left blob is not a single round ball. It has a large dense base, which is cluster 0, a separate arm reaching upward, cluster 2, and a small populations tucked inside, which is cluster 4 and 5. The arm shape of cluster 2 is meaningful. It is not floating freely. Um, it is connected to cluster 0 at the base. So these are probably related cell types that split along one axis of variation. What else we can find? So the middle blob, the elongated shape, is connecting two larger masses, clusters 1 and 7. This elongated shape is a classic sign of a continuous gradient or differentiation axis. So cells are not discretely in one state or another, they transition gradually. Cluster 3 is sitting in the bridge between cluster 1 and 7. This will tell us that predicting protein values for bridge cells will be harder because they don't cleanly belong to one pattern or another. And we have cluster 6, which is completely isolated, far right. Um, this must be a cell type that is fundamentally different transcriptional program from all other cell types in this dataset. So that's it for RNA UMAP. In the next video, we're going to overlay protein expression on RNA UMAP. So thanks for watching. Hope you enjoyed it.